So for today, our main topic is probabilistic methods in phylogenetics, and the other important thing included in the lecture notes for today is quality control of phylogenetic trees and phylogenetic inference. If you look at Moodle in section phylogenetics, you'll find the new lecture notes as phylogenetics tree probabilistic methods. We'll talk about phylogenetic models a little bit. This is kind of background information for bioinformatics in general, showing how simple things in creating models might help you in improving your algorithms and simulating biological events with fairly easy mathematical models. We don't go to mathematics just show what kinds of parameters we may need for reproducing evolution and nucleotide sequence changes. And then the important topic of quality control comes last, but before that we'll discuss the probabilistic methods of maximum likelihood and Bayesian phylogenetics. We need to go through some of the topics quite quickly, but that will leave us some time to do a demo of a maximum likelihood tree and look at some of the parameters in Mega, which we didn't discuss yet. Now, this is a repeat of a previous slide talking of the classes of phylogenetic methods, and in bold-faced we'll see probabilistic methods which we'll discuss today, and then the reliability testing. The probabilistic methods are interesting in that instead of focusing at a distance matrix of sequences, they are looking at the total sum of the multiple sequence alignment, which is the data, candidate trees, which we try to find as the optimal ones, the very best trees, and also the theory, which is included in the tree search, and that includes the parameters. And in maximum likelihood, the question is basically set finding the best tree that would have the highest likelihood that we would observe an alignment given the tree and the model. So we are looking at the kind of a conditional probability of the data given the theory. So let me repeat it again. It's a weird and complicated concept, but even if it's a short sentence, but we are trying to maximize the likelihood of observing the alignment given the tree and the model and its parameters. And then this proceeds as a tree search in the way we discussed, trying to sample the tree space and find good candidate trees and seeing if we get any improvements over the changes in the trees. So we have our tree generator, which is swapping branches. We get a new tree, we get a score for the tree from the scoring algorithm, and if the score is improved, we start with the new tree to generate another one, and if the score is worse, we go back to the previous tree and make a new one. Now, I'll try to do a quick introduction in the models of sequence evolution. The models have to account for many things. For instance, the base composition in the organism, whether we assume that all the bases are present at 25% frequencies or different from that. Then we know that in real biology, not all mutations happen with equal rates. There are transitions which are changing a small base to a small base or a large base to a large base, keeping the size the same for the nucleotide. And then we have transversions where the large is changing into a small or vice versa. And the transitions are much more common and that they should be accounted for. Then we have heterogeneity of evolutionary rates in at least in all protein coding sequences. There are, for instance, within the triplet, we have the last position in the codon, which is free to change without changing the protein sequence. And even if we look at the non-synonymous substitutions, we have important sites in the protein where the amino acid needs to be conserved, or if we look at the MSA, we see that they are conserved sites. And then we have other sites where we have room for variation. And within the tree, we may have different rates in different lineages, or in other words, different rates at different parts of the tree. So assuming that we are looking at a large tree with fishes and land-living tetrapods, and we look at enzymes involved in gas exchange, like carbonic anhydrases. So the evolutionary pressures would be very different in fishes living in water and having very widely differing concentrations of oxygen and CO2, whereas in the atmosphere, carbon dioxide and oxygen are pretty constant, at least for the physiology of breathing. Of course, in the cells is different, but just as an example of why some lineages might have different rates. Phylogenetic models are really simplistic in the sense that they don't care about the biology. They are just looking at strings of letters which represent our DNA or amino acid sequences. And they are treated as Markov models, and I need to skip the explanation here. And the real mutation mechanisms or any features of natural selection need to be ignored. They are not part of the models, but we can devise some parameters that might simulate it, or we can have some ways of allowing for different rates in different sites, as here in the third point. And the models can't really give us real numbers of actual rates of changes, how many nucleotide changes in 1,000 years or 1,000,000 years or in a generation or anything. So we only have the relative rates between the taxa in our data. But still, with all these limitations, the way our tree algorithms can find phylogenetic trees, phylogenies, which seem to be consistent with everything else we know about evolution and speciation. So it seems the algorithms work quite well, so that we have the features we need represented by a handful of parameters there. Okay, now the transitions have a higher rate of mutation because when we have two very large bases, purines with two rings in the base, they collide and they create a disruption in DNA structure, and therefore they are very easily fixed by DNA repair mechanisms. And likewise, if we have a transversion creating a base pair or attempted base pair of two pyrimidines, which are the small bases, a single 6-membered ring, they don't even reach each other, so that we could have the hydrogen bonds. And again, this is an obvious problem in DNA structure and will not be missed by proofreading mechanisms in DNA replication. Whereas in case of transitions, the small to small or large to large mistakes might be missed by the proofreading mechanisms more easily, and therefore more of them are created in the random process of DNA changes. But anyway, no matter what the mutational mechanisms are, whether it's blaming the DNA replicase or whether it's blaming radiation or whatever, so we can just give a high probability to transitions, that is changes of A to G or C to T or vice versa. Okay, now if we think about the changes, we have like 16 different changes of states in a Markov chain that is going through different nucleotides in a single position of a sequence in a single column of an MSA. We have the probabilities of the nucleotide staying the same here in the diagonal, and then probabilities for every change. This could also be called a rate matrix. And in the very simplest model of evolution used for phylogenetics, it is assumed that every change has an equal probability, and then you would only need a single parameter mu for the overall substitution rates, and then every rate of change would be mu divided by 4, and we don't need to care what is the probability or rate of things changing the same. But then if you assume that transition frequencies are larger, so we can have a parameter kappa, which is the ratio of transitions and transversions, which would be larger than 1, and we just set the transversion rates into 1 and the transitions into kappa. So these are the CT and AG changes. Just going back here, so you can see that CT changes are here and AG changes are there. But then you might think that maybe these transitions are not quite equal to each other. So if we have a lot more of AT base pairs than we have of CG base pairs, which is true for the human genome and for any warm-blooded animals, because having 50% of CG would be too hard to separate. The CG base pairs are stronger with 3 hydrogen bonds instead of 2 for AT. So when we have more of CT, so then it means that, sorry, when we have more of AT, it means that the A to G mutations happen more often than G to T mutations, even if they have the same probability of happening for an individual base. So we could start thinking of taking the nucleotide frequencies into account to make a correction for that. And the simplest version of that is just giving the nucleotide frequencies here. But then if you mix it with the principle of having transitions as more common, so we get the HKY model. The reason I'm presenting these models here is that you have a short form of presenting what these abbreviations mean, because you find them in the selection of different models to choose from in the phylogenetic program. So you might remember some of these, or you can at least refer back to these notes when you figure out. And I can tell you that the simplest model is not really of any use, so you can skip Duke's Cantor in any case. And then we have some more complex versions where you allow the transitions of large to large to be different from the transition from C to T. But then going to the most generalized version, in which you allow each change to be with a different probability and each base frequency to be different. So we have a total of 9 parameters, 6 different substitutions, and 4 for base sequences. Well, we don't have every rate to be different, because this is assumed to be time reversible, so that we have the same probability for going forward and backward in a change, and with that we get maybe we need to go forward. What I wanted to say is that we have the bottom half of the matrix as a mirror image of the top half of the matrix, so that the probabilities of change are reflected, and therefore we have only 6 parameters for substitutions, and then there are 4 for base frequencies. But anyway, the real message here is that it's a good idea to accept GTR model if you don't need to do any, if you don't want to do any further model testing or estimation of what is the most appropriate model. For nucleotide sequences, this is not a problem. 9 parameters is not a lot, considering that the data sets are often hundreds or thousands of nucleotides, meaning hundreds or thousands of characters. But for proteins, when we have a 20 by 20 matrix of amino acid changes, so and 20 different amino acid frequencies, that comes up to hundreds of different parameters. And considering that we have just 1/3 of the characters in the protein alignment, that would mean that we might have the ratio between data and parameters in the order of just a single digit number. So it could be like 3 or 5, and that is certainly too highly parameterized. It is overfitting into a model which is too complex to the amount of data we have. And so for proteins, we have nobody is considering the GTR model, but we have a couple of simpler models to try, which we'll see in Mega. And if you do model selection, which is testing your data, whether it has equal frequencies of nucleotides, whether the alignment shows transitions and transversions happening at equal rates, whether we have C to T and A to G transversions happening at the same rate, and so on. So you could get away with the narrower ones, but the benefit of choosing another model is considered to be minimal nowadays, so that you might even skip the part of model selection. But if you see model selection discussed somewhere, it is because it used to be a big deal, so that people wanted to be sure that they use the simplest possible model, which is still appropriate. But anyway, now you have the abbreviations in the preceding 7 or 8 slides, so you can refer to them if you need. And the real thing to remember is that the general time reversible model, GTR, is what you need. Okay, let's go back to probabilistic phylogenetics. We have the maximum likelihood methods which try to maximize the probability of observing the alignment we had given the tree we are studying, which we have found in the tree search and the model for the evolution. And the results of ML are considered pretty good because it is a robust method, so that even if we have some flaws in data, so there might be some sequences which are missing parts, or we might have minor misalignments, it's still okay. So it's tolerant to noise in the data. And the branch lengths you get with ML are supposed to be accurate, and you get likelihoods for the sites, and also you get the ancestral states for the positions in your MSA. So, or you get ancestral states for each node in your tree corresponding to each position in the MSA. So as the result, you can reconstruct the ancestral sequence at some ancestor nodes, like the last common ancestor of human and mouse, a primitive vertebrate. Okay, the bad points of ML is that for proteins, the kind of theoretical and biological basis is not there. When you are looking at jumps from one amino acid to another, it might require one or more nucleotide changes underlying it, and they are not really accounted for. It has been tried, but it doesn't work. If you just count the number of changes, you are not getting good trees. And the results are quite dependent on the model you choose. We'll see the different protein models in Mega. And also it's computationally intensive, so that the models need to be quite simple, and often you don't have the time to compute fully resolved large trees using 5 different models and compare them. But anyway, the good sites are considered definitely overriding the bad sites, so that you can create publication quality phylogenetic trees with maximum likelihood methods. But because there are people who are against using ML for protein sequences, so you might be more advised to use Bayesian phylogenetics for proteins, or convert your protein sequences to the corresponding DNA sequences when you do ML. Okay, moving to Bayesian inference. This is a nice comparison of the approaches taken in maximum likelihood phylogenetics and Bayesian phylogenetics. So when maximum likelihood is trying to maximize the probability or likelihood of data given the theory, so the Bayesian inference is looking at the posterior probability of the theory given the data. So we are reversing the approach. The probability that the model, and that means the probability of the tree and parameters being correct given the multiple sequence alignment, which is our original data. And this is the famous Bayes formula, which you may have seen in your high school mathematics book, given just with letters A and B for two events. And here we are looking at a theory which is labeled with the theta here, the theory including the model which contains the tree and all the parameters and collections of rates and having possibilities for different rates across sites and so on. So all these parameters and the tree are incorporated in the theta, and this looks kind of deceptively simple, but this is what you get, and with that something you try to optimize. But the problem is that there is no analytical way, there's no mathematical way to get numbers for this. And the reason for that is that the probability of our data is not available, and if we try to compute it, it would be huge multidimensional integrals, and we would need to, it's just not computable. And even theoretically, considering the probability of a multiple sequence alignment, it's philosophically very hard to define. So what we do is we just try to do a simulation that is getting to approximations and trying to see what kinds of trees, what kinds of values for the parameters are most frequently visited by the simulation, and use that as a proxy for the posterior probability. So instead of, in the same way as you could try to do a mathematical evaluation of the probability of coin tossing and figure out that if it's a flat object which never falls on the sides, you probably get heads and tails both 50% of the time. But instead of this analytical procedure, you could do a Monte Carlo simulation of tossing the coin over by 1,000 times, and the more times you flip, the closer you get to getting the true probability is 0.5 for each outcome. And in the same way, we can observe the outcome trees, how often particular clades are included in the trees, and get the posterior probabilities by simulation. Okay, the method of simulation is Markov chain Monte Carlo, and once we have a long simulation with lots of trees, we sample from them, not maybe every step of the simulation, but every 10, every 100 simulation steps, and then we get the probability distribution, and from that we'll see the posterior probabilities of parameters and trees. And let's see, I probably skipped the heated chain idea. And I probably need to skip even the stochastic mechanism, but let's think about the simulation in a metaphorical way. The algorithm explained here is telling us that as soon as we are improving in the probabilities, so we just keep going ahead. But then if we are taking a step down, we make a random choice of a number which we can accept, a drop in probability which is acceptable, and the choice is such that we will have smaller probability of accepting large drops and a higher probability in accepting smaller drops. And this allows our algorithm to be climbing over the hills in the probability landscape and hopefully reaching the global maximum. But whatever the case, this algorithm makes sure that most of the time is spent in the high probability areas, walking around in small distances, and the system of heated chains allows us to, and chain swapping allows us to go into other mountains not visited by our chain, but maybe the other chain in the simulation. But in the end, if we allow the program to do the initial hill climbing from the random initial position, and we ignore the first few thousand steps until we are in the good probability region, this is called the burn-in of the simulation. When we ignore the burn-in and just look at the stable area of the simulation, we can then sample the results and get the posterior probabilities for our parameters and trees. It's kind of miraculous and maybe not so easy to grasp with a 2-minute explanation, but I'm just advising you to take my word that this is a good and valid method and gives us good trees, and this is generally accepted as one of the state-of-the-art methods of phylogenetic inference. And the MCMC might be quick even if we allow for 100,000 steps, if we have a small alignment, not too many taxa, not too long alignment, so we can have it in a few minutes. And if we have a real-life problem of hundreds of nucleotides, hundreds of amino acids in the alignment, and maybe 20-30 sequences in the taxa, so it would mean that we might need to run for several hours before we can be convinced that our run has converged, that it has had a long enough time for spending the sampling in the best regions. And I probably need to do a Mr. Bayes demo to make these concepts a little bit more understandable. Okay, so Mr. Bayes is a software I'm not recommending for beginners. Beast is a better one. And because of the shortness of time, this is the last time we can discuss phylogenetics. I need to just record short demos of Mr. Bayes and Beast to get you to see how they work. And maybe if you need phylogenetic methods or really want to practice, you can learn more on your own. But this is like just helping you for the first steps in your journey in phylogenetics, in Bayesian phylogenetics. And is Bayesian phylogenetics good? And the short answer is yes. There used to be some debate because it doesn't have anything related to biology or mutational mechanisms. Instead, we are just simulating the parameters and letting the simulation decide our model. And as conclusions of the probabilistic methods, so both of these model-based phylogenetic methods are considered to be state-of-the-art or the best known methods at the moment. And Mr. Bayes has been probably the most popular for the past 5 or 10 years. But you have to remember that because the computation takes a long time for each tree, so or each iteration, if you think about maximum likelihood and bootstrapping, which we'll discuss in a minute. So you might not have sufficient computational time to sample a large enough fraction of the tree space if your data set is too large. So it would mean that you fail to find the best region in the tree space before the run is over. And because of that you really need to take care of your quality control to see if the tree is reliable. The quality control numbers will reveal if you have not sampled your trees sufficiently or if there was a problem with the original data. And let's move to reliability testing. So bootstrapping is something you can do with the distance methods with maximum parsimony and maximum likelihood. The idea is that you resample your data set. So let's say that you have an alignment of 100 columns. So you resample with replacement, which means that you first take a random column of the alignment and you put it in the control data set. Let's say we get number 7. And the next time we take another column from the set, we probably get something different, but towards the end we have a high probability of getting a second copy of something we already have. And once we have 100 columns, we probably never got exactly one copy of every column in the original alignment. So we get 0 or 1 or several columns. So it is similar to our original alignment, but if you remember that the order of the alignment in this tree building effort has no meaning. So each column is considered to be a separate character, and it doesn't matter in which order they are in the genome or in which order they are in the amino acid chain of the protein. But in bootstrapping, you take a random simulated data set which contains a different number of the characters you had, different numbers of the characters you had. And it means that if you have some columns in the alignment which are not reliable or which are kind of misleading your tree building. So if you leave them out, you might get a different kind of a tree. But anyway, if you do the bootstrapping, repeating it 1,000 times and then build a tree out of each of the bootstrapped data set. So you can then compare the trees and get frequencies for each of the clades. You get support for the clades in the tree, and you can build a consensus tree which shows the most popular clades kind of consolidated in a single tree. And this is indeed a tree. You probably need to watch that in the original instead of my tiny screen. I need it to expand into full screen as well. So now the numbers here over each node represent the support for the clade in the bootstrap data set. So having 99 here means that 99% of the simulated trees included a clade which has has the three taxa, coelacanth, human, and mouse, CARP11 from each. This 99 does not really care about how these three are ordered, but the internal order can be seen here, that the human paired together with mouse as the closest pair is supported by 100% of the bootstrap trees. And you can see that there are some nodes here which have a bit more uncertainty, so going to 67 or 58%. So maybe in this node we might have some trees where this Capitella carp was actually the fourth one in a clade at this level. But these percentages show you how well a clade which is under or like radiating from this node, how well supported that clade is. In Bayesian phylogenetics, we don't do bootstrapping because the simulation is really long. You would need to do a new run of maybe several hours for getting new optimal trees, and you can't repeat that 1,000 times. But instead, we get the posterior probabilities directly from the simulation. We can get those probabilities even for the clades. And the algorithm is optimizing the posterior probability of the whole tree given the model and the data. Oh, sorry, given the data. So it's optimizing the probability of the tree and the model given the data. So we get the posterior probabilities for the clades, and we get probabilities of how likely our final parameter values are going to be as the most appropriate ones. And the posterior probabilities are values of between 0 and 1, and in bootstrapping it's considered that 50% is a nice threshold for accepting a node. We talked about binary trees being simplified into flattened trees when we can't be sure of the order of some nodes, so flattening some nodes into these comb-like shapes. So 50% is a typical threshold for bootstrapping, but 0.5 in posterior probabilities is thought to be weaker support, and there's no agreement what should be the value for flattening nodes for Bayesian trees. Some people use 0.5, some people use 0.75, some people go as stringent as 0.9. But definitely anything which has a support of 0.9 or higher is a very highly supported node in a Bayesian tree. And we don't have Bayesian trees here, but we saw some in the previous data sets, in the previous lecture notes for some carbonic anhydrase research of mine. But let's talk about method selection. This might be the most important slide here for future reference. If you start a project and you start collecting sequences, you probably need to figure out what are valid sequences for including in the study, and it might be a good idea, of course, to do the MSAs and remove sequences which look broken, which don't fit with the others, which are database errors or incomplete sequences, not having all the exons in the genome or whatever. But we can also do do sequence selection or taxon selection based on initial trees. So we can use quick methods for inspecting trees of 100 or 200 sequences. And we can see if there are some serious outliers which have very long branches. They might also look bad in the alignment, being very, very different from the others, considering that the branch length represents the estimated distance. And then in the highly accepted methods where we can only take few dozen sequences, we can use our initial trees to break down the problem into smaller pieces. So let's assume that we have 10 subtrees which are really robust and have high bootstrap values in neighbor joining or minimum evolution trees. If we accept these subgroups and take just, let's say, 2 representatives of each subgroup, we can then do a Bayesian tree with 20 sequences. And if we have the 10 subgroups, each with 10 or 20 sequences, we can then make separate trees for each of the subgroups within themselves. And then we can use the upper level tree, which has the representative sequences, and use tree building software to include our separately computed subtrees into the kind of backbone tree which we made from these representative sequences. So there are many ways, and obviously you have problems in that approach, in that because they come from different runs of the algorithm, you can't necessarily compare the branch lengths between separate trees. But anyway, this is a nice approach to get a good quality plausible topology and good bootstrap values for various nodes. And now for model selection, I'm starting to disrespect the model selection more and more, having read articles where even others say that it doesn't really improve your results. But we have a link to a program called IQ3, which does maximum likelihood, including model selection. But when I've done a couple of tries, I was always getting use the GTR model and allow different rates for different sites. And that's basically what you can do even before doing model testing. And so just some more advice before I conclude this part of the lecture. It's important that you have controlled data. If you have garbage in, you're obviously getting garbage out. If your sequences are not homologous or if they are misaligned, the trees are not going to be reliable. So review the data and do some common sense inspection, look at the alignments and see if something seems to be out of place. Most of the time you have lots of sequence data to select from, so you can easily drop bad sequences and just work with the sequences that align well, sequences which don't have long intervening sequences, etc. And sometimes even the worst methods give you the same results as the best methods, and that happens when you have a clear phylogenetic signal. If you have sequences which do not go into very large distances so that you would have the risk of misalignment, and if you have a nice collection of different levels of identities of sequences having higher similarities and lower similarities to each other over an even range. So that's something that helps in the tree structure. And if you have multi-domain proteins where you can infer from the annotation that you have clearly separate domains or parts of the sequence which code for a globular part of the protein sequence. So analyzing a single domain at a time is advisable. You might also have quite different mutational rates for two different domains, making your analysis more complex. But the higher risk is the idea of exon shuffling, which might bring different non-homologous domains into multi-domain proteins which share one homologous domain. And so if you analyze a single domain at a time and make sure of the homology of those domains, so you are good. And then the use of nucleotide sequences is recommended in phylogenetic inference because then you have more data to work on. You have the silent mutations or synonymous substitutions to give more differences between cases where the protein alignment would have an identical amino acid. So looking at very highly conserved sequences like CA-related protein 8, I've made protein trees where I get a nice rake-like shape for 15 sequences out of 20, and then 5 which are going into a tree-like shape, and that obviously didn't work. So I had to go back to DNA alignment. But it is not a good idea to do multiple sequence alignments based on DNA because the risk of misalignment is larger, and also when you have your gap insertion algorithm, you might be accidentally inserting gaps of 1 or 2 residues, and that would certainly mean that you are comparing non-homologous nucleotides because the frame shift caused by an insertion of 1 or 2 guarantees that your translated amino acids represented by those alignments would be very different. So you really need to do protein alignments, of course, provided you are studying protein coding sequences. But whenever possible, use proteins for alignments and then use some software. How2null is something we link in the course to substitute the original coding sequences into the protein alignment. This is abbreviation for protein alignment to nucleotide alignment. And why is it a bad idea to do a reverse translation directly from protein to DNA without considering the actual mRNA sequences? Right. So it's common to make a conceptual translation of a protein coding mRNA sequence or a cDNA sequence just using the genetic code. But why is the reverse not such a great idea? We could get the theoretical DNA sequence from the protein using the genetic code, but what is the catch there? Well, one codon can represent one amino acid. Exactly. We have biologists in the audience who know the idea of ambiguity in the genetic code. It is not ambiguous when you translate codons to amino acids. Each codon has just a single translation, but the back translation may have several options. Most commonly, it's 4 different ones. So you would have to do guessing, and you don't get the actual sequences. So you have to find the cDNA sequences from the databases, and often it's easy enough. The coding sequences might be given as crosslinks in the protein entries, so you get them easily. Or at the very worst, you need to do a blast search with your protein sequences to find the real cDNAs. But anyway, once you have those alignments, so it's easy enough. If you use Ensemble for retrieving sequences, it's fairly simple to get the corresponding protein sequences from the database. And likewise from RefSeq, it works really, really well. But anyway, this is what I wanted to say of probabilistic phylogenetics, and let's do a small demo with Mega and look at the parameters we didn't discuss.